Everyone, today's my topic is immobilization technique, which we'll discuss further. So let's begin. Im- Immobilization technique is basically imprisonment of a cell or whole cell in a distinct supporter or matrix. In next slide, we will discuss the supporter molecules. So supporter or matrix allows exchange of medium. It means if a cell is movement from one medium to another. So medium basically consists of three substances like substrate molecule, effector molecule, inhibitor, receptor. First immobilization technology was used for the amino acylase in Japan by using bacteria Aspergillus. Okay, the matrix or support which is used in the immobilization techniques, it has certain characteristic. So first one is it's hold the substance which has to be immobilized from one medium to another. Second, it must be cheap or it should be easily available. It's a wide range of metrics which we used in this immobilization technique. Basically, there is a reaction between takes place between the enzyme or the cell and the matrix which hold the enzymes or a cell for the immobilization of the from one medium to another. So the site of the adsorption, or we called the reaction where the takes place, that is the support molecule where the cell adsorb itself by means which the immobilization take place. Matrix are grouped into three major categories. First is natural polymer, second synthetic polymer, and third one is inorganic material. In natural polymer includes like starch. Starch is a good high water holding capacity it has, and the alginate is also derived from the algae cell wall. Synthetic polymer, it include for the ion exchange resin or polymer, like it is immobile insoluble support with the porous surface, and in these pores the cell is incorporated when the immobilization takes place. Examples of such synthetic polymers are like polyvinyl chloride, UV activated polyethylene glycol. Third is inorganic material, that includes silica, glass, charcoal, activated carbon, and zeolites. The classification of the cell immobilization techniques, it is basically divided into two parts. So you have to clear here that the enzyme immobilization and the cell immobilization both have the same mechanism by which they immobilized. So there is a classification which is based on the physical bonding and the chemical retention. It means the bond between the supporter molecule and the substrate, that is cell or we can say enzyme. First is adsorption, second is ionic bonding, and third is covalent bonding, which comes into chemical bonding, and fourth that is entrapment, in which a semi permeable membrane is formed around the enzyme or cell particles. Fourth one is entrapment, in which agar is used, which has a pore having large sizes, in which the cells or the enzyme particles settle down and the immobilization take place. And in next slide we can see the diagrammatically representation of different mechanism of the immobilization of the cell. Thank you.